Hello students, welcome to Lab 8. We're going to be talking about the movement of molecules. We kind of talked about this during lecture at one point, but we are going to be talking about this for lab. So, the movement of molecules across the cell membrane is an essential part of homeostasis. Plasma membrane plays a large role in controlling the movement of molecules from one side of the membrane to the other. So this is what we talked about as passive transport or active transport. So passive transport doesn't require ATP, active transport does. In today's lab, we'll be studying two major forms of passive transport. This is diffusion and osmosis. There is a difference between diffusion and osmosis. So diffusion is the tendency of molecules to move from where they're highly concentrated to where they're less concentrated. This is just in a solution. So diffusion is from high concentration to low concentration. So if you just have a solution, if it's evenly distributed, that's fine, it's equilibrium. But if you have just an area of high concentration and an area of low concentration, you're going to have some diffusion to that low concentration area. So a few substances like oxygen and carbon dioxide can diffuse across the plasma membrane or just in the air, right? If you have oxygen and carbon dioxide in the air, you're going to have some diffusion going on. So if I breathe out, that's going to diffuse, right? You're going to have some diffusion in the air of that carbon dioxide. Osmosis is the term to diffuse water or other liquids, right? You're going to have some type of liquid diffusion across a boundary, across some type of boundary. So like any molecule, water will move, or liquid will move from high concentration to a low concentration. So living cells contain large amounts of water or various solutes dissolved in it. So water is the solvent, and then you'll have a solute dissolved in that solvent. Water is most likely going to be a universal solute or solvent. Say I get confused in that all the time. So water is mostly going to be a universal solvent. As the concentration of solutes increase in the solution, the concentration of water decreases. So you have a bucket of water, and as you increase the solute, that concentration of water is going to decrease. So diffusion is the movement of solutes. You can control that movement if you can control the amount of solutes in that solution. So you can also say that air is kind of a solution in a matter of speaking, if you want to call that as a liquid container. So diffusion is the movement of substances from an area of high concentration to an area of low concentration. Many molecules can diffuse across the cell membrane. It doesn't always, but it can diffuse across the cell membrane. Tonicity. So tonicity is the term used to compare the water content of two different solutions, right? So we can use this term to describe what will happen to a cell if the concentration of the solutes is not equal to each side of the membrane, right? So you have the outside of the membrane and the inside of the membrane. Most of the time you can't control what's inside the membrane, right? But you can control what's outside of the membrane. most solutes cannot diffuse across the membrane, right? You can't control the solutes diffusing inside the membrane because it's too big, right? But you can control what happens to the water that can go inside the membrane. So we can only study the movement of water across that membrane. So first, the experiment on the baggies with the starch and iodine. In this experiment, the baggie represents a semi-permeable membrane. The baggie contains starch, or the baggie contains iodine. So when the baggie is placed in the beaker with the iodine, when it has a starch in it, the iodine diffused through the baggie and reacted with the starch, turning the starch blue-black. And I have the graph right next to this page. So what's in the baggie? It's the starch, right? What's in the beaker? Just iodine. So the baggie, the starch is in there at the beginning, but at the end, it's both iodine and starch. So what moved into the baggie? It's the iodine. And by what process? Diffusion. So this is the setup. This is what you're going to write in your notebook. So with this setup, you can answer all the questions afterwards. And you should be doing that. Okay, so on your assignment, on the page that I give you, you can answer all those questions. The next experiment is osmosis through the movement of water. We have 10% sodium chloride solution. That's the solute, and water is the solvent. So the movement of water through the potatoes, water moves in or out of the cells by the process of osmosis. So this is osmosis, the movement of water through those membranes of the cells. So we have the hypotonic solution. Water will move into the potato, making it feel turgid. This is through the distilled water, right? Hypo, meaning it's going to feel bigger, and then hypertonic, meaning it's going to plasmalize. It's going to feel smaller. So potatoes, slices in salt water. This is the 10% sodium chloride solution. So water will move out of the potato, meaning that the slices feel limp or floppy. Okay, once again, this is the results of the potato osmosis experiment. You can write this down in your notebook. Also answering the questions after that. Okay, the next experiment. We have blood cells put into distilled water. 0.9% sodium chloride and 10% sodium chloride. So water will move out at 10% sodium chloride, meaning they'll lose water, and then there'll be no effect on the 0.9% sodium chloride, and then they'll gain water at the distilled water, meaning they'll be hemolyzed. This is the results of that experiment. So tube 1 is 10% sodium chloride, tube 2 is 0.9% sodium chloride, tube 3 is distilled water. So we can see here, when red blood cells are in a hypotonic solution, they're hemolyzed and will appear clear red. And that's in A. When red blood cells are in a hypertonic solution, that's in 10% sodium chloride, they're crenated. Right? They'll burst and they'll appear cloudy red. And that's in tube B. After that, we have osmosis in plant cells. This is the allodias. So when they're in a hypotonic state, they're more turgid. When they're in a hypertonic state, they're plasmolyzed. We kind of saw this in the potatoes, but we're going to see this more closely in the allodia leaves. So here's the results of the allodia experiment. And if you can see here, the chloroplasts are all filled. The cells are all filled with the central vacuole. It forces the chloroplasts away from the center, and the cells are all turgid. Okay, that's what's supposed to be normal. Now, if you filled the slide with 10% sodium chloride, this is what happens. The chloroplasts are pulled away from the center, pulled away to the center of the cell. The plasma membrane is pulled away to the center, and this is what's called plasmolysis. And you can compare this between them two. You can see differences where the cell walls are on the sides, but everything else is in the center. So the cell is turgid, it's hypotonic, and water is flowing into the system by osmosis. If the cell is flaccid, it's been placed into an isotonic solution. Water flows in and out of the cell at the same rate. It's fine, but at this point it's kind of wilted. If the cell is plasmolyzed, the inside has been shrunk. It's a hypertonic solution. Water flows out of the cell. Cell begins to shrink. It causes the plasma membrane to tear away from the cell wall. Okay. Make sure to answer all those questions on the lab assignment. Here's some review for lab practical number 2. I will try to get to the lab practical number 1 as soon as I can. There is a lot that I have to grade so far, so give me some time on those. Okay, if you have any questions, please ask me and let me know, and I will see you later.